Hello everyone. Today this is the third lecture of module 2. Today I am going to discuss about translation, how the information present in mRNA get decoded by a complex machinery. So that is the essence of translation. So mostly I will restrict myself here mostly by discussing overview of protein synthesis and the concept of genetic code and the mechanistic details of translation. It is a very complex process, but I will mostly highlight fundamental concept about translation and some insightful ideas regarding this translation process. So here translation means the synthesis of protein inside our cell. So as I was discussing about replication as well as transcription before in the previous classes, so DNA molecule which actually contain the information like in terms of nucleotide sequence, and then it get converted into RNA, and this process is called transcription. And DNA goes to DNA, that also I mentioned before, that is replication. Now, in both cases, whether we are synthesizing DNA or we are synthesizing RNA, in both cases we are basically synthesizing polymer of nucleotides, where the nucleotides are attached by phosphodiester bond, and the basic chemistry of DNA or RNA synthesis, they are basically same. But now here the next step of central dogma, we will be getting proteins. So here protein molecule, and this process is called translation. Now, if you remember, when I am discussing about replication or transcription, we mentioned their enzyme, the major enzyme involved in these processes, like DNA polymerase, enzyme that catalyze or that polymerize deoxyribonucleotides. Similarly, when we are getting RNA through transcription, the enzyme is called RNA polymerase, the polymerase which polymerize ribonucleotides triphosphate. But in that case, translation, like logically the name should be protein polymerase, but there is no protein polymerase. So translation is happening, or I would say translation is being conducted by a big machine that is called ribosome. Ribosome helps in process of translation. Now, in this case, in protein, amino acids are linked by peptide bond. So let's learn what is peptide bond and how it is formed. So how amino acids are actually linked together, this is the question. So since proteins are polymer of amino acids, first I should little bit introduce about amino acids. So if we see amino acids, so this is the basic structure of amino acid. They have one amine group and one carboxyl group, and here you have the side chain and another hydrogen atom attached to the central carbon. So now that's why this is called amino acid. Both the amine and the carboxyl group, both the acid and amine group, both are present there, amino acid. So now we have 20 different types of side chains present. Like for example, if I put here R1, this is one specific type of side chain, and that should be designating a particular amino acid. But now how these amino acids are getting attached together by peptide bond, that I am going to show now. So if I draw the structure in this way, so H, N, H, C, R1, H. So this is the same amino acid, right? And now another amino acid, if I draw in this way, just in nearby amino acid. So this is a defined amino acid. I am putting R2 as the side chain. So if R is just a hydrogen atom, then it should be glycine. It's the simplest amino acid. So now here COOH. So if those two amino acids, they react together and they make like something like this structure here, H2N, C, R1, H, C, O, and this OH and this H from the next amino acid, they can live as water molecule. So as a result of that, what we are getting here, N, H, C, R2, H, COOH. So now you can see these two amino acids are now attached together, and this bond, this is called peptide bond. I discuss so many things about phosphodiester bond when we discuss about nucleic acids, but this is peptide bond, and by which 20 different types of amino acids naturally present in our body, they get attached and they make a specific type of protein molecule. Now, one important thing is to make peptide bond in lab chemically. To make peptide bond is not very simple job. So chemically, if we need to make peptide bond, we need multiple organic solvents and also much higher temperature compared to our body temperature. But if you see in our body, inside our cell, the temperature is much lower, like ambient temperature, body temperature, and also we have aqueous solution, not organic solvent, but still everything is happening properly. Protein synthesis is constantly happening, and that's why we are healthy and happy. So let's learn how this synthesis happens. This is a complex process, but I already told I will stick to the very basic and fundamental aspects of protein synthesis. So translation, the overall concept. Translation means in protein synthesis inside the cell. That's all. And here, if I start from the beginning, so I'm just writing something here. Say for example, if I write something here. So something just written using English alphabet. Now if we know how to read English, if we learn the English language, then we can actually decode the information. Similarly, in our cell, inside our genetic material, that is DNA, so information are written in such a way. But in this English literature, English language, I would say, there are many alphabets, but in our gene, we have just 4 alphabets, A, T, G, and C. And just the combination of that, it will make some kind of sense and we will get protein. And that is the functional molecule, and that will be doing function like everything happening in our body. So whatever written here, if we know English, then we can try to read this. So I can just intentionally I didn't give any space, anything, because in our gene, in our DNA, those nucleotides are present like that, just continuously it's present. So here if I just break them just after 3 letters. So now we are getting some kind of meaning that the sun had set but was not out yet. So similarly, if we decode the information present in gene, so finally, like in the intermediate stage, it will make the RNA, particularly mRNA, the messenger RNA, and then after translation, we will get some meaningful product. Just I am showing here the sun had set but was not out yet. So also I intentionally use only 3-letter word. So 3-letter word I used. In living system, those nucleotide sequence, those are also get read by just 3 nucleotides consecutively, and those 3 nucleotides present consecutively, and they mean something that is called codon. So I will explain in more details. So see, if I write here something like this, some sequence here, say A, G, C, U, G, G, A, U, G, A, G, G, C, 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 A, and we can put many sequence here, and then for example U, A, G, C, and then 3 prime. So this is our 5 prime end and 3 prime end. So what is this? So this is nothing but some mRNA sequence, messenger RNA sequence, just I am putting to explain the process of translation. Now as you can see here, here this is mRNA and it should be decoded by ribosome, as I already mentioned, the major machine which make protein, that is called ribosome. So as a result of that, when ribosome will bind to the 5 prime end of this mRNA, it will sit on the 5 prime end of mRNA, and there is some kind of signaling sequence, some specific sequence. There is little bit variation in eukaryotes and prokaryotes, but I will discuss that little later. But ribosome will sit there and start reading this sequence which is present in this mRNA. And while the ribosome is, so this is ribosome, right? Ribosome. When the ribosome is moving in this direction from 5 prime to 3 prime direction, then whenever it will encounter the first AUG, this is only AUG, right? First AUG. So if I start from 5 prime end, it's written A, G, C, U, G, G. So then you can see this AUG. So whenever it will get this sequence AUG, from there the protein synthesis will be started, and this is called start codon. So these 3 nucleotide letter together we say codon. So this is the start codon. Start codon. So whenever ribosome will find the start codon, after that every 3 nucleotides will be another codon. It's just going like this. And what is the function of codon? Codon will code for a specific amino acid, that's all. So codon will bring some amino acid in the machinery, where in the ribosome, where the translation is happening, and there will be some catalysis reaction, and finally we will get the protein. So as a result of that, AUG, the start codon, it always bring one amino acid, is called methionine. So we have 20 different amino acids. So methionine is the starting point of a protein. So AUG is the start codon. And I must mention here, this kind of code is very much universal. So methionine or the AUG, like AUG should be the start codon in every mRNA. during every protein synthesis. Not only that, for every organism, whether it's bacteria, whether it's plant or animal, that doesn't matter. The start codon should be AUG. If you ask me why, I don't have the specific answer. I would say this is because of a million years of evolution and life chose this thing in this way. That's all. So AUG is the start codon. Then the next amino acid, for example, the codon here is AAA, so it's code for lysine. So you don't need to remember which which codon code for what amino acid. It's not required. So we generally use some kind of table that is called genetic code, by which we can understand which codon refers to which amino acids. Then because of the GGC, we will have glycine, another amino acid. Then CCA, it will code for proline, and it's going on like that. So ribosome is basically adding amino acid one after one based on the sequence present in mRNA. That is the catch. That is the main thing, the translation. So you are making a specific product which is a polymer of amino acids based on the mRNA sequence. And the mRNA sequence actually came from the DNA. So as a result of that, the actual information present in DNA, but by several steps, we are making the product which is a protein. And when ribosome is moving in this way from 5' to 3' direction, whenever it will encounter this codon is UAA. So this is stop codon. Stop codon. So then no matching amino acid will come. So in this case, the protein synthesis will stop. So protein synthesis will stop at stop codon. Okay. Now I would like to discuss little bit more thing about this genetic code and codon. So I'm just mentioning few codons here. So how many codons should be there? So this is very simple, very logical answer, that how many codons should be there, because as I already mentioned that we have just 4 nucleotides and 3 nucleotides together makes a codon. So if you do different types of combination, then you will get how many? 4 to the power q, right? So that is 4 into 4 into 4 equal to 64 codons present. So this is the total number of codons present in living system. Just 64 words in that way. So now out of this 64 codons, there are 3 stop codons. I just mentioned here one UAA. Apart from that, 2 more stop codons are UGA and UAG. So those 3 are the stop codons, and start codon I already mentioned AUG. So as a result of that, those 3 stop codons, they don't specify any amino acid. But now if we just ignore those 3 codons, then we have 61 codons left. So now those 61 codons, they are responsible for how many amino acids? Because I told naturally in our cell we have 20 different types of amino acids. So they code for 20 different types of amino acids. I have given few examples while I was explaining here, for example, methionine, lysine, glycine, proline, etc., and their corresponding codons. So if we have 61 codons and 20 amino acids, that means some kind of degeneracy is there. That means I would say multiple codons actually specify a single amino acid. In few cases, multiple codons specify like a single amino acids. So that's why this is the this number relation, 61 codons and 20 amino acids. So whatever I mentioned until now, this is a very, I would say, methodical and simplistic way of how ribosome read the sequence present in mRNA and it makes protein. But the actual scenario is much more complex, right? So that's why, so if you see the complexity of biological system, so it's really complex scenario. So if I start a little bit of that, the first question here is how a particular amino acids they are coming because of this AUG. For example, I told that AUG code for methionine, but why? How the methionine comes? Similarly, AAA, it brings lysine in the translation site, but how it will come? So that is the major question. So, and that time when all those things were getting discovered, it was a really a challenging question. And that time, many scientists, they used to thought that those 3 nucleotides present in a codon, maybe they are making some kind of chemical pocket, and that chemical pocket can attract the side chain of a specific amino acid. But it is really hard to imagine how this will bring 60, 20 different types of amino acid, how this process is happening. So actually in this scenario, again, Francis Crick brought some hypothesis. So he suggested something that is called adapter hypothesis. So adapter hypothesis means Crick was suggesting that maybe inside our cell we have some kind of smaller molecule, smaller I would say polynucleotide, like some kind of nucleic acid molecule, which may act as adapter. Like all of us know what is adapter. Sometimes we use adapter to charge our devices, for example, laptop. Similarly, what Crick was suggesting that if we have some adapter molecule and that is made up of nucleotides, then it can recognize the sequence present in mRNA, the codon sequence it can recognize, and by different method it can bind to a specific amino acid, so that that particular adapter can bring a specific amino acid in the translation site. Remember, when he proposed this idea, that time tRNA was not discovered. tRNA stands for transfer RNA. It was not discovered. It was just hypothesis. But scientists found those small RNA molecule that is called tRNA, and that actually brings amino acid in the site of translation. So if you see here now, so this is, I'm just some cartoon diagram of some tRNA. So tRNA, transfer RNA, it is also polymer of ribonucleotides. Now if you see, this is made up of nucleotide sequence. So this is one loop. So this is one loop present in this tRNA, and here you have complementary sequence to the codon. So for example, in this tRNA, the sequence will be U, A, C. Why? Because that's why UAC actually recognizes AUG through Watson-Crick base pairing, to hydrogen bonding. So as you can see, UA and triple bond here. So this way, this tRNA can recognize this particular codon, and this particular tRNA can bind to a specific amino acid, that is methionine. And that's why whenever you have the codon AUG, it will always bring the amino acid methionine. That's all. Similarly, next tRNA, it will bring another amino acid I already mentioned here. So it is bringing lysine. So the sequence in the loop should be U, 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 so that they can recognize this codon sequence AAA present in mRNA. Now this loop is called anti-codon loop. Why? Because it binds to the codon sequence present in mRNA. So anti-codon loop. So codon anti-codon recognition, and that is actually dropping the correct amino acid at the site of translation. And then some catalysis is happening. But let's see, in the next slide, I'm going to discuss a little bit. I will add something more here, but this is the basic schematics, basic explanation of protein synthesis, how it is happening. So here, the translation, the involved machineries and the processes. So as you can see, the template for translation or the template for protein synthesis is definitely, all of you know that it's mRNA. And the major machine I mentioned is ribosome. And ribosome is made up of RNA and proteins, many proteins involved to make this ribosome particle. And then we have tRNA, those adapter molecules, transfer RNA that brings amino acid at the site of translation. Now see what is happening. And also if you see this ribosome, we have, in ribosome we have 30S, we have 50S, those are the different subunits of ribosome. In case of this is ribosome from E. coli or bacteria, and they have 30S and 50S subunits. Similarly, in case of eukaryotic system, they have 40S and 60S subunit, but those details are not required to understand these fundamental aspects of protein synthesis. And now, if I put this sequence particularly here, this is an mRNA sequence, and if you see whatever I explained, the first AUG should be the start codon, and then it will go like this. Then after AUG, every 3 letter will be bringing some specific amino acids, and we are getting polypeptide. So polypeptide and protein, they are very similar thing. Polypeptide generally we say when the length of this polymer is much smaller, and when it is much bigger and it has some kind of 3-dimensional conformation, we generally refer that as protein. Otherwise, they are same in terms of chemically, if you say that amino acids are linked together by peptide bonds. Now, if you see how ribosome will actually bind to this mRNA. So here, for example, in case of bacterial system, they have some specific sequence at the 5' end of the mRNA, and that will attract ribosome. Ribosome will get attached to this site, and this is again some kind of orth-centric base pairing, because I already mentioned ribosome is made up of RNA and proteins. So that means whatever the ribosome binding site I am mentioning here in this sequence, for example, 5' UAAGGA. So some complementary sequence is present in some ribosomal RNA which is present inside the ribosome, and by virtue of that, the ribosome will come and bind here, and then it will move towards the 5' to 3' direction, and then the first AUG, whenever it will encounter, then it will start protein synthesis. So as you can see here, this is the ribosome. It is attached in this sequence now. It just recognizes this sequence, and now it will move from 5' to 3' direction like this, and the polypeptide or the protein will be synthesized. Here I am mentioning that nascent polypeptide. That means the polypeptide is just being synthesized, nascent polypeptide. So in bacterial system, I mentioned this kind of sequence present, which is called ribosome binding site. Similarly, in eukaryote, when I was discussing about the processing of eukaryotic mRNA, I told that there is some kind of cap, special cap called 7mg cap present at the 5' end of eukaryotic mRNA. So in eukaryote, this 5' 7mg cap is recognized by ribosome, and it will help in translation. Now, next question is how a specific amino acid should be attached with a specific tRNA, right? So tRNA, they are bringing us bringing the correct amino acid at the site of translation. So in this slide, I am trying to explain that. So this is the tRNA, you can see here, this is tRNA, and this is specific for phenylalanine. Phenylalanine is an amino acid. So the system is, that for specific tRNA, we have some enzyme present in our cell. In this case, I would say this enzyme, that their generic name is amino acyl tRNA synthetase. So they will add a specific amino acid with the tRNA. So in this case, the amino acyl tRNA synthetase specific for phenylalanine. So as a result of that, for other tRNA, they have some other enzymes and they will add a specific amino acid to that tRNA. So you need some energy in order to carry out this reaction, ATP here, and then finally you will get amino acyl tRNA. This is charged tRNA, sometimes we say. So amino acyl tRNA. So where the phenylalanine, this amino acid here, this is the amino acid, it has been already added to the tRNA. And this tRNA will go and bind to the mRNA sequence during the translation. As you can see in the mRNA sequence, now if you have the UUU, this is the codon, then AAA present in this anticodon loop, and this is our codon now. So as a result of that, this phenylalanine is coming into the site of translation. So this process will continue, and as a result of the correct sequence of amino acid will be added just based on the sequence present in mRNA. You should always remember that. That's why we are actually translating like from the mRNA template. And here some informations are given which I already discussed about genetic code. So as I already told, it's very difficult to remember which codon code for what. So here this is the genetic code table here. So all 64 codons are like genetic code like codons are mentioned here and their corresponding amino acid. We don't need to remember that, but just for an idea. And everything we discuss is briefly mentioned here. And as you can see, this is a fundamental step protein synthesis mechanism, and this is same, this is universal everywhere in plant, animal and bacteria, as I already mentioned. And because of this process, Hargobind Khurana, Nierenberg and Holly, they got a Nobel Prize in 1968 for their beautiful work on protein synthesis. And here I would say some stimulating discussion is still remaining here. I would like to explain this thing, because if you go into much more details about translation, like in textbook, you will get like 50 pages of different types of discussion there. So many factors, so many protein molecules, so many things are involved to make this process more efficient. But the basic logic I already explained, how protein synthesis is being occurred inside our cell. But now the question is, if you see this central dogma here, protein we are basically getting at the end. This is the last step of this central dogma. But now, before that, we need to replicate our DNA, and then we have to make the RNA through RNA polymerase. So, but if you see this DNA polymerase or RNA polymerase, all are proteins, right? So they are enzymes, but they are basically they are proteins, protein enzymes. So as a result of that, the question is that how those proteins, how those enzymes are formed. If the protein formation is at the end, how life started? So this is kind of a difficult question to answer. This is kind of I would say this is if you say it's like this is a chicken and egg problem. So this is egg and this is chicken egg problem. Because if we don't have the protein, then how we are getting this RNA polymerase, DNA polymerase, and not only DNA polymerase, RNA polymerase, I mentioned some other enzymes also. All of them, they are protein. So as a result of that, the idea is that RNA has enzymatic activity. So RNA can perform some enzymatic activity. So this is like this can be discussed in somewhere else also, that RNA world hypothesis. RNA came first, because as I already mentioned in some other classes that RNA can be genetic material also. As you can see in some cases, in some virus, they have RNA as their genetic material, and it can also catalyze some reaction. So as a result of that, RNA has the enzymatic activity. So maybe at the beginning of life formation, maybe during that time RNA was the genetic material, and that can act as genetic material as well as that has some kind of, they have some enzymatic function also. Similarly, if you see the ribosome, which is the major machine for protein synthesis, so ribosome, and ribosome is made up of rRNA, ribosomal RNA plus so many proteins. Many scientists work for long time their structure function, like structural and functional relation of ribosome, how they catalyze the reaction, so many things. And interestingly, they found this catalysis, the protein synthesis is like is performed by rRNA, not by protein. So rRNA actually acts as an enzyme. See, this is actually acting as an enzyme, RNA. Particularly, we say PTC, peptidyl transferase center. So this is the region of ribosomal RNA that catalyze the reaction. So particularly, I would like to mention here, see, this is so fundamental step, that protein synthesis, but it is being catalyzed by some non-protein enzyme. Protein is not involved there. So as a result of that, again, it actually suggesting that RNA world thing, that so fundamental thing, the protein synthesis is happening in every second, in every life form, but it is still being catalyzed by ribosomal RNA, or you can say just RNA, but not by protein. So because of their beautiful work, not just these three scientists, many other scientists actually contributed a lot. So in 2009, Venki Ramakrishnan here and Tom Saik and Ada Yonath, so they received Nobel Prize in 2009 in chemistry for their beautiful work, particularly understanding the structure function relation of ribosome and how it helps in translation and so many different types of insights of ribosome. Now, this is the last slide here. So, translation machineries, that means particularly I would say ribosome, it is an attractive target for therapeutics. Particularly, if you see many of the antibiotics, antibiotic means some kind of drug, like medicine, what we generally take to prevent bacteria. So antibiotics inhibit bacterial growth. So now if you see many antibiotics, for example tetracycline, so this generic name in commercial, like for example some doxycycline, tetracycline, so those antibiotics, they actually target ribosome. So that binds to particularly tetracycline and doxycycline, that binds to 30S ribosome and blocks the binding of amino acyl tRNA to the A site. Now the thing is, this translation is happening in both bacteria as well as human, and I told it's fundamentals, fundamental step, fundamental process, but still there is some differences. And so many scientists actually, when they worked on detailed understanding of ribosome, that actually elucidated that what are those minor differences. So based on that, if scientists, they can design some antibiotic which will specifically stop the protein synthesis of bacteria, so then those are the antibiotics, like they are targeting ribosome, but they are not creating problem of eukaryotic ribosome, or I would say particularly human ribosome. So as a result of that, they are very good medicine to prevent bacterial growth. So if no protein synthesis is there, so bacteria will not survive, they will not divide in our body. So we have more example like chloramphenicol, which is present in many of our eye drop. So that also prevent the peptidyl transferase reaction, that means this peptide bond formation on 50S ribosome. And then streptomycin, erythromycin, those are some other example of antibiotics. There are many more, but they actually block different stages of translation process. And this is one, I would say, some interesting question. You may try to look into it. What could be the possible answer? That why ribosome is an attractive target for the development of antibiotics. Because if you like try to buy 100 different types of antibiotics from a medicine shop, you will find many of them, like many, they are actually targeting ribosome. But what's the reason for that? So that's all. So you can follow any of these textbooks. Like all books are very good. So any textbook you just feel comfortable, you can go with that. Thank you very much.